fifth session. This session is about the cancer regulation and immunomodulation by diet. Here we have four distinguished speakers. One and last one is Gökhan Hotamışlıgil. He will connect us with virtually. First, I will introduce Sebastian Brandhorst from Southern California, United States. Dr. Brandhorst is a research associate professor of gerontology at the University of South California, Leonardo Davis School of Gerontology. Dr. Brandhorst earned his PhDs from the University of South California and University of Duisburg, Essen. Dr. Brandhorst's training is based on vast background in cell biology, molecular biology, medicine, and biochemistry, all of which are highly relevant for my focus on biogerontology research to identify the mechanism underlying health and lifespan regulation and their translation into the clinical application. He has performed research related to aging, lifespan, and age-associated disorders, particularly cancer. Subsequently, he focused on the role and application of short-term starvation, low-calorie and low-protein diets, so-called fast-mimicking diets, on health and lifespan in preclinical and clinical setting. Please, Dr. Brandhorst.
Thank you so much. Good night. Thank you for the introduction and thank you for the invite. It's really been a pleasure and it's been a great workshop so far. I'm really excited to be here and share some of our results. And I think, I know this is a session focused on cancer, but I know Claudio is going to do a boatload of cancer, so I'm going to instead give you an overview over sort of the work that we have been doing in the lab at USC with regards to fasting and subsequent a fasting mimicking diet that we developed in the lab, and sort of cover a lot of the basis with regards to overall health and lifespan, but also for some of the treatment and prevention of some of these aging-associated morbidities. So with that, I want to point out I am technically a gerontologist. My interest is in understanding how organisms age, but also sort of really relay the message that aging really is the catalyst of chronic diseases. So understanding how the system fails and the aging phenotype, how that really relates to these typical aging-related diseases is of really interest to me. And with that, also the idea that if we learn how to optimize the speed and the quality of aging itself, this could really be a powerful intervention to increase health span. So the idea for me is not necessarily to live longer, Chris asked earlier, but I think the idea for me is more to really increase the quality of life, and that really relates to the idea that we want to minimize the time that we live, that we spend with like a poor or just average health, right? And so this idea here on the slide is really that I want to highlight that decisions that we make early on in life can put us sort of on a trajectory that rapidly decreases our health, and with that we see an onset of sort of stress, fatigue, a number of sort of high cholesterol, and then really comorbidities that make us live with a number of diseases for an extended period of time. Whereas we have the option and really the choice that depending on what we do across our health or lifespan, we can actually be fairly active, fit, with great cognition and overall health really easily up into the 60s and 80s. So the idea really is here, can we identify approaches that maybe put us back from this poor health towards a more healthier, longer life. And while I acknowledge that there's a number of approaches you can investigate to get you to this reversal, our focus on the lab is really focused on diet and nutrition. And the idea, obviously, to use dietary approaches to improve health and lifespan is nothing new. Caloric restriction has been around for almost a century. You already heard that some of these studies being discussed. So caloric restriction on top. We had time restricted eating that was mentioned earlier. Approaches that Michelle commented on, intermittent periodic fasting. And when I started working at USC, we used a lot of water-only fasting approaches where we essentially gave mice only access to water for about 72 to 96 hours. Mice would lose about 20% of their body weight during that time. And we had very promising results with regards to cancer treatment. But the problem with that was that these severe sort of fasting approaches were greatly limited by translating that to the clinic. You would have a hard time telling a cancer patient that they're going to fast 120 hours, right? Most oncologists would not really support that. So we got a grant from the National Cancer Institute to develop a diet that has fasting-like properties while providing some sort of nourishment. And here the idea really is to reduce the carbohydrates and reduce protein. Instead, we're buffing that up a little bit with fats, but we're still a calorie-restricted diet. So this is really what I wanted to focus on the rest of this talk. Why dietary approaches? So one example is here, and we heard about this earlier already. So these evolutionary preserved nutrient-sensing pathways are really a unique target to delay the onset of aging-related diseases and aging itself. So when you look across these species here, yeast, worms, flies, and mammals, and essentially color-coded, you can see that these pathways, these intracellular pathways that respond to nutrients are preserved. So, for example, you're a target of rapamycin, PI3 kinase, and so on. And the important thing is that you can shut these pathways off, and you can do this by restricting the abundance of nutrients. And what that does is turning cells off. So when nutrients are abundant, cells will continue to grow and happen. If you take their food away, they essentially stop growing, and they're now entering a cellular mode in which cellular resistance is increased. You can test it fairly easily by giving cells high doses of chemotherapy. Once you fast these cells by withholding their macronutrients, these cells actually can endure higher doses of chemotherapy without dying. So we had a grant that we submitted about a year ago, and one of the comments that came back from one of the reviewers was essentially, do you really think that your diet can just cure everything? And I thought that was weird because I believe diets can do a lot of things. And you can see this sort of summarized here, that at the center of all these essential functions really is the idea that what we eat and when we eat matters. We can really turn on and deactivate these pathways, and all of them ultimately are linked to some sort of cellular function, whether it's resilience, tissue repair, organismal function, and metabolic homeostasis. And we also know that once these systems fail, that leads to the onset of many of these diseases. So yes, diets are essentially so powerful because all of these pathways are targeted. And I think that makes them more interesting than a simple drug, where it's, say you want to, for example, use a drug to target AMPK, right? So you would essentially act only here, whereas the diet really offers you a unique opportunity to look at a number of functions. So the fasting-mimicking diet that we developed, just to give you guys a brief overview, is plant-based. It's focused on natural ingredients. We developed it in the lab, and we have done a number of clinical studies on this now. We have shown that this diet is effective in trials looking at aging and multiple chronic diseases. It's a periodic reduced caloric intake. So the idea here is we, in mice, for example, we give this diet for 4 days, and then we allow the animals to completely, without any further intervention, regain all their body weight. They can eat whatever they want for the next 10 days, and that way we repeat these cycles. The same idea in humans is it's a 5-day intervention. So the first day is about 1,100 calories. The subsequent days are somewhere around 650 to 800 calories. It's a sort of a meal plan. The patients would get a pre-labeled box for each day. They're supposed to consume only what's in these boxes. That's usually including some sort of soup, nut bars, olives are in there, teas, and they're really not encouraged to consume anything else. So So it's convenient, but it's restrictive. So people get a little tired of eating these. It's low protein and low medium complex carbohydrates and high in fat. The first time we published data on this is this cell metabolism paper here. And then this is essentially focusing on decent mice with a small human cohort. And then in this published in Science Translational Medicine, this larger study focused on in the clinical trial. And I'll show some of these results. So essentially what this fasted thinking diet does, it reduces the number of markers that we routinely look at with regards to particular focus on sort of like anti-cancer response, and that's including the reduction in fasting glucose. So when you compare these 72 hours of water-only fasting in mice compared to 4 days on the fasted thinking diet, you see similar effects with regards to fasting glucose, namely 40 to 45% reduction. With that, we see about a 10-fold increase in ketone bodies, a reduction in the sort of like pro-growth stimulating hormone insulin like growth factor 1, and we see an increase in the IGF binding protein, which essentially inhibits the bioavailability of IGF. So the effects are comparable, and then all we did is we gave this to mice, starting in middle-aged female mice, when they were about 16 and a half months, and put them on this diet every other week, essentially. And so what we saw across the board was markers indicating a multi-system regeneration. You can see, for example, in the slide here that in these old mice, at this point they were 20 months old, liver weight, when you put them on the fasted intermittent diet, shrinks, the liver dramatically reduces in size by about 35% or so. But when you refeed these mice, that's the gray bar next to it, sorry, right here, within a few days, the liver completely regains all its weight. And we looked at that in detail, and it's really an induction of sort of like stem cells within the liver that wants to initiate refeeding, start to re-expand its tissues. You can see similar effects here with regards to the white blood cells. So you can see obviously age-dependent decrease in white blood cells. These are animals that were maintained on the FMD, but immediately after the refeeding, you can see these levels of white blood cells in 20-month-old mice are comparable to what we would see in about 6-month-old animals. We see a overall reduction in tumors within these mice. The idea here is that they do not develop different tumors, they just have less of them. And we see, I guess, less spreading of these tumors, so less organs are affected. You can also see that this diet reduces adiposity. So in red here is a cross-section across the visceral adipose tissue. The control mice here on the left, you see substantial fat deposits compared to the fasted intermittent diet fed animals, less with obesity. And then when you look at survival, there's an interesting point here where we have about an 11% increase in median lifespan, but towards the end, these older animals actually don't do as well. And it's a discussion Michelle and I just had. I think that is really indicating that we're still at a point where we don't really understand the age-dependent nutritional requirements that come into play. So this diet is pretty severe, and these animals, again, once they were really old, had a hard time regaining weight from this diet. So there was really no benefit in being here. Until then, they did pretty good. There was also indication of an increase in the immune and cognitive rejuvenation. So I'll show some of the examples of that. But with that being said, this is sort of overall health span evaluation. We did a number of studies that looked at can these dietary approaches prevent the onset of aging-related diseases, or can we use it as a treatment modality? The first one that I want to point out, which is the most sort of common approach for us, is using this diet as a assistant with regards to cancer management. And what you can see here is essentially these top two graphs is a mouse breast cancer model, 41, and the bottom here is a melanoma. The difference between the top and the middle panel is a different drug. So this is cyclophosphamide, this here is doxorubicin. So these are two drugs commonly used in fighting breast cancer. And essentially what you see here in black is the regular progression of these tumor cells. They're implanted subcutaneous into the flanks, so you can measure growth. You can see that once you give them this cyclophosphamide here, it does what it's supposed to be doing, namely reduce tumor progression. But similarly, just 3 cycles here with this fasting mimicking diet was as effective in reducing tumor growth as chemotherapeutic agent, and most importantly, combining the fasting mimicking diet with the drug significantly reduced tumor growth. And this works for the other models, and Claudia will comment a lot more on this in the next presentation. I'll leave it at that. I'm going to show you an example here of a mouse model for type 1 diabetes. What we do here is we essentially induce hyperglycemia by giving them this drug STZ. What you can see here at the bottom is a staining of the pancreas, and in red you would see the insulin-producing beta cells. With STZ, these insulin production is essentially eliminated, leading to a increase in blood glucose levels over time. And at this point here, all these animals actually have died from type 1 diabetes. What you can see is also in light gray here that just by putting these animals on the fasting mimicking diet for 4 or 5 cycles, reestablishes sort of normal blood glucose levels, and that is because we induce sort of the reintroduction of beta cells within the pancreas, leading to insulin production, and therefore the animal survival. Another model that we use is a high-fat diet-induced obesity model for cardiovascular disease. And I'm just showing the lifespan here, but what you can see immediately is that giving mice a high-fat diet induces these little butterball mice. They're really big. They're probably somewhere in the 60 to 70 gram range compared to mice in the mid-20s. So these are control mice. These are mice just fed with a high-fat diet. And then here you can see these mice were maintained on a high-fat diet, but given access to the fasting mimicking diet every 2 weeks. And so this pretty much normalized their body weight and almost helped them almost recover from this high-fat diet-induced sort of early death, right? This light blue curve here, they're coming back closer to the control. With that, we saw also a reduction in these cardiovascular disease phenotypes that is induced with a high-fat diet. Lastly, example where we use the FMD with regards to Alzheimer's disease shown here. So these are essentially EFAD mice. These are a mouse model for Alzheimer's disease that we used. And when you look here within the subiculum, quantified here at the bottom, you can see the A-beta load. You can see in red here the animals that were maintained on a fasting mimicking diet had a significant reduction of A-beta within the subiculum and the cortex. So in mice, we know that this diet is increasing health and lifespan, and it reduces the risk for the development of age-related diseases. The problem really for us was how do we successfully translate it, and how do we measure if it is actually feasible. So the problem with using chronological age, so meaning your date of birth to sort of evaluate if there's an improvement in physical function is that you can't really change when you were born. So you're never going to change the chronological age, and that we're all aging pretty heterogeneous. So for example, all these people, you say they're born in 1984. The reality is they're probably more biologically different. So here the idea is somebody could be living a very healthy life, but biologically probably closer to a 37-year-old, or it could be living unhealthy life. You might be chronologically 40, but your body is resembling more of a 43-year-old, right? So the importance of that is that your biological age is a much better predictor of the onset of diseases for you. So we use an approach to measure biological age in a cohort that I showed you before, the short summary on, and use this to sort of evaluate whether or not that our dietary approach can improve their biological age. So here again, we try to reduce this disease and potentially increase lifespan by applying a dietary approach to put us back on a healthy age trajectory. So this trial was developed or designed as follows. It's essentially a crossover trial, so meaning the arm that was in the fasted mimicking diet at the beginning was then maintained on sort of their regular diet, whereas people that were on a control diet were allowed to go into the fasted mimicking diet arm. So they crossed over, so everybody did the FMD. In this case here, again, it's a 5-day intervention followed by 25 to 26 days of regular food intake. We did that for 3 consecutive months, and we had a baseline evaluation. We had an evaluation immediately after the first fasting mimicking diet cycle, and then a sort of refeeding evaluation where after the 3rd cycle, these patients were already back on their normal food for about 5 to 7 days, and we did the same measurements for the control arm. Okay? Important to note that at baseline, there were no significant differences between the 2 arms. You can see here with regards to male and female, they're somewhat evenly distributed. Average age is about 42 and a half years. They're fairly comparable with regards to education. And then the BMI, also fairly evenly distributed. We had people sort of in the normal range, overweight, and then we had obese range. So just to really make the point that we think that this fasted mimicking diet has fasting-like effects in humans. I'm just highlighting the same parameters that I showed you earlier for mice, namely fasting glucose, ketone bodies, IGF-1 and IGF-1. So we see the same trend. In humans, it's a little less dramatic than in mice. Mice have a really high metabolism. If you fast a human for 3 days, they will not lose 20% of your body weight. So results that we see here is, namely after 3 cycles now, a significant reduction in BMI, and that is not based on a loss in lean body mass, but it's coming from a loss of fat mass. Also, need to mention glucose levels here at this point are not significantly different, but again, at this point of this measurement, they have already been consuming normal food, so you wouldn't expect them to have lower glucose levels. The sort of more interesting data came from a post-hoc analysis, and in this case here, we just stratified our patient group based on whether they are at risk for certain markers. With regards to BMI, you can see the average loss in BMI was highest in those people that were essentially obese. You can see that people that were in a pre-diabetic range, namely blood glucose levels being higher than 99 milligrams per deciliter. In this case here, these subjects, after these 3 cycles on FFB, returned their blood glucose levels back to normal levels, essentially. And then you see other effects, systolic, diastolic blood pressure, IGF-1, etc., all affected. So indicating that this dietary approach seems to be most beneficial in those people that sort of need an intervention. So then, briefly with regards to biological age. So in this case, we used markers that were developed by a colleague of mine, Morgan Levine. And in this case, we're using sort of a multi-system approach where we take serum markers. We don't use epigenetic spots in this case. We just use these markers here, albumin, alkaline phosphatase, creatine, C-reactive protein, HbA1c, systolic blood pressure, and total cholesterol. And so all of these get fed into a model, and that model essentially then calculates whether or not a person is, or what the age of this person is biologically, right? So you put it all together and then compare it to a larger cohort, the system will be like, yeah, these markers make it resemble a 37-year-old, but you're 40, so in that case you would be biologically younger. You could also go in and say, I'm 40, but all my markers make me look like I'm 54. In that case, it would be considered an accelerated. So with the FMD and with Morgan's modeling, what you can see here, first of all, this is the control group. So all these dotted lines are essentially the baseline change, median decrease, and sort of a modal decrease. So you can see that in the control group, there really is no change. At most, they have a small shift to the right, indicating that they have aged. Whereas on the FMD group here, you look at the change in biological age. So The 0 here would indicate no change, but we see a shift to the left, so indicating that after 3 rounds with the spasticity, there's a reduction in biological age based on the markers and the serum markers that we measured. So you can, if you're optimistic, you could say something like over 3 years, more like me, maybe say a year and a half. And then I want to make it clear, the next slide is hypothetical modeling. It's just running numbers on a simulation, but to take it with a grain of salt. But the idea here is, if you were to tell a 50-year-old to do 3 cycles of the spastic mimetic diet for the next 20 years, what would happen? So again, it's a simulation. Just based on the outcomes, when you look at their life expectancy at age 70, you see this dramatic increase of the FFG. So people would live longer. You can see this also here with regards to life expectancy. The red in all the graphs is the spastic mimetic diet compared to the people that would not do an FFG intervention for 20 years. So you see an increase in life expectancy, a significant reduction in all-cause mortality, heart disease, cancer, cardiovascular disease. The actual percentage is not here. So again, this is modeling, so be cautious about it. If you want to look more into this, we just published this in Nature Communications maybe a month ago. So that's all I have. Here are my conclusions. So obviously, diets, including other lifestyle factors, are a powerful tool to prevent or at least delay the onset of any chronic age-related disease. These cycles are effective in reducing risk factors for diabetes, cancer, and cardiovascular disease in clinical studies. And given that the risk for most major chronic diseases rise exponentially with biological age, slowing of the aging process could lead to prolonged disease-free life expectancy by delaying the onset of age-related chronic conditions. And I want to point out that the data that I showed you with regards to biological age was not just based on one study, but we replicated it with the second study. So this seems to be, at least across those 2 studies, similar. And that's all I had. Again, I want to thank you for your attention. Just hold our data, and there are any questions.
Thank you very much. As you see in these caricatures, you see how do you, what is your guess? How we can change your results from benchmark to bad? I think so. It is not easy to change the daily life of the population.
Well, I think first of all, diet is a habit, right? It's something that we have created carefully over time, or more or less carefully. But I think there's also a lot of social influences on what we eat and how we eat. And I think that is something that can be cautiously modulated. Again, Michelle and I just talked about it. I was like, these fasting and fasting diet interventions are, generally speaking, pretty severe. To tell you to not eat for 5 days, you will probably do it if you are extremely desperate. So if you have already disease indication, you might be more inclined to try it. But I think that there's other dietary interventions that and exercise that are actually easier to adhere to for a longer period.
Thank you. Do you have any question? Please, Michelle.
There's quite a lot of people that seem to get worse with the diet, the biological age. So do we know anything about those people, and is it age-related or?
So the short answer is we do not know. We looked into that with Morgan. So there is within this total cohort that we looked at. I have to point out that the overall average of these people that was in the study, they were already biologically younger than what they chronologically should be. And I think that is sort of like an enrollment effect that healthier people tend to be more inclined to do these sort of things. So what exactly drives this increase in biological age in a small subset of these people, we were not able to identify. We looked at contributions of BMI, CRP, all individually to see if there is any factor that drives it. The answer is not. We also shown, we see that these effects here are not driven by BMI. So it's like if you correct for that, you still see these effects. So the answer is we don't know. Mm-hmm. And I think that is a very interesting question that I want to tease out. But we're running into issues of essentially cohort size at that point. Mm-hmm. By the time you look at the handful of people that gets older, then we don't have the power to sort of dig through that. So we would need a larger study.
Thank you.
Okay, Claire.
I can.
Congrats, great talk. I have one question regarding your model of aging in animals subjected to high-fat diet. So do you necessarily need to initiate the fast mimicking diet very early to reverse the vast majority of the negative effects of high-fat diet, or there is some chances for people, for mice, and potentially also for people who are obese at middle age to reverse the process and to prolong their expected lifespan?
So for the model that I showed, the animals were young, the idea for that is it was pretty unhealthy mouse diet. The idea was more to initiate sort of cardiovascular disease, because you would have a hard time doing that in mice otherwise. So yes, that was severe. So in this case, the testing when we sort of disrupt these negative phenotypes with the diet wasn't really the aim. The data that I showed you on that larger panel, those were middle-aged mice. We have done it a couple of times in cancer studies in older mice. Still works, but it's a little less effective. So I think there's for sure an age effect, but I think that you see that across the board in humans too, that doing something is better than doing nothing. So I think there is a lot of opportunity in doing something even if it's
Thank you.
Okay, thank you very much. Let's go to second speaker, Claudio Vernieri. Thank you very much. Claudio, Dr. Vernieri is the group leader of PIRC, Institute of Molecular Oncology in Italy, and works as a medical oncologist in the breast unit of that institute, Nazionale dei Tumori. He directs the metabolic reprogramming in solid tumors program, and his research specializes on clinical and translational research activities on cancer metabolism and the design and conduction of clinical trials aimed at assessing the safety, feasibility, and anti-cancer activity to specific metabolic interventions such as calorie-restricted diets and metformin. And he graduated with honors in medicine and surgery at the University of Federico II of Naples, and then obtained his PhD in molecular medicine in Andrea Cliberto Laboratory. Dr. Vernieri, his title, Nutritional and Pharmacologic Metabolic Interventions to Program Cancer Metabolism. Please, Dr. Vernieri.
So, thank you very much for the nice presentation, for the invitation, and thanks to you and for organizing this meeting in memory of an excellent scientist and a great man, who was J. Mitchell. So, as anticipated today, I will tell you something about what we are doing in our hospital to translate some of the metabolic approaches that have emerged as potentially effective against cancer, including calorie restriction and metformin in cancer patients. So, as you probably know, metabolic reprogramming has been recently included in the list of the hallmarks of cancers. So, it is one of the most fundamental properties that allow tumor cells to grow, to proliferate, invade, metastasize, and escape the immune system. And basically, there are three types of interventions that three classes of metabolic treatments that can be reprogram cancer metabolism. Among this one, we are mostly focused on dietary or nutritional interventions, which are based on the assumption that by changing the type and amount of foods and nutrients that we introduce through our daily life, we can change the concentration of crucial metabolites in the blood and in tumor microenvironment. And in particular, in the last years, we have been focusing a lot on calorie-restricted approaches, including cyclic fasting or fast-mimicking diet, which, and the use of these approaches in oncology is based on this quite simple model that comes from a huge amount of preclinical literature published the last 30 years. We are now working on this model that was originally proposed by Walter Longo, and we will come hopefully soon with, let's say, an enhanced model. But basically, you can see here, it is very well explained how the vast majority of normal cells react to nutrient starvation, in particular to glucose and growth factor restriction, by slowing down their growth and proliferation pathways. And this allows them to enter a sort of protected mode, which allows them to be protected when they are challenged with cytotoxic compounds. On the other hand, the vast majority of tumor cells, which are driven by oncogenic pathways that stimulate the unrestrained growth and proliferation, these tumor cells are unable to stop their growth and proliferation even in condition of glucose, growth factor, and nutrient restriction. And so they go on growing, proliferating, trying to synthesize new molecules, and when they are challenged with cytotoxic compounds, they are unable to repair damaged structures such as DNA, but also proteins, lipids, because they do not have sufficient energy, and also anabolic molecules. So that said, this is the outline of the presentation. Let's very briefly start with the preclinical evidence supporting the use of fasting or fast-mimicking diet in cancer therapy. This has been in part anticipated by Sebastian. In particular, in this paper that was published in 2012, the group of Walter Longo at USC demonstrated that combining different types of cytotoxic compounds with water-only fasting results in synergistic anti-tumor effects in different tumor models, and also regardless of the specific cytotoxic agent that was used. Not only this, but in specific models, the authors also demonstrated in a very clear way the protective effects of fasting on normal tissues. In this experiment, you can see that this is the volume of tumors from animals treated with control conditions, fasting alone, which is in this case is not effective against the tumor. Then there is here a line which you cannot appreciate because it is the line of animals treated with a high-dose chemotherapy, particularly high-dose doxorubicin, and all of these animals were dead just a few days after treatment initiation because the treatment was too toxic. And in the red line, you can appreciate how mice who were treated with high-dose chemotherapy in combination with cyclic water-only fasting were all alive at the end of the experiment, and the tumor growth was almost fully controlled. So this picture summarizes basically through experimental data the model that I showed you before. So, but as anticipated by Sebastian, the use of water-only fasting in the clinic is unfeasible for the vast majority of cancer patients, and this is the reason why in the last decade several authors, starting from Walter Longo's group, proposed different approaches collectively referred to as fast-mimicking diets. This definition comes from a recent international consensus that was led by Andreas Mikkelsen in Berlin, and also Walter and me participated to this. And you can see that fast-mimicking diet is any diet that is specifically composed to induce the metabolic effects of fasting while allowing for a potentially higher caloric intake, including solid foods. It usually refers to a plant-based, calorie-restricted diet with a maximum of approximately 1,000 kilocalories per day that lasts between 3 and 7 days. And in this paper published in 2016, the authors demonstrated that cyclic fasting mimicking diet is as effective as cyclic water-only fasting, here indicated with STS or starvation, in terms of slowing down in vivo tumor growth and also in synergizing with cytotoxic treatments. And in recent years, different laboratories, including our group, contributed to demonstrate which are the metabolic determinants of cyclic fasting mimicking diet anti-cancer effects. For instance, in this work that we published in 2021, Giulia Salvadori demonstrated that glucose restriction in murine triple-negative breast cancer models is responsible for the reduction of the cancer stem cell compartment, and glucose restriction is the only metabolic mediator of the anti-tumor effects of fasting mimicking diet, because it is sufficient to add back glucose to fasting animals to fully reverse the anti-tumor effects of the FMD. In the same paper, Giulia also demonstrated that the vast majority of more differentiated tumor cells is not affected by glucose restriction and fasting. Not only this, but these cells activate escape pathways such as the PI3 kinase AKT-mTOR pathway or CDK4 or CDK6 kinase to basically escape the anti-tumor effects of fasting mimicking diet. Indeed, combining cyclic FMD with a pharmacological blockade of each of these axes resulted in a remarkable prolongation of animal survival, and in particular in this experiment with the cure of the vast majority of these animals. Totally different story applies to hormone receptor-positive breast cancer, which is the most common breast cancer subtype. In this paper published 2020, we demonstrated that endocrine therapy synergizes with cyclic fasting or fasting mimicking diet in several mouse models of hormone receptor-positive breast cancer, but in this tumor type, glucose does not play any mechanistic role, because all the anti-tumor effects of calorie restriction are mediated through the reduction of blood insulin, insulin-like growth factor 1, and leptin. And indeed, it was sufficient to add back each of these growth factors or a combination of them to in vitro culture tumor cells or to fasting mice to reverse the anti-tumor effects of fasting. And in particular, in this experiment in which we combined endocrine therapy plus CDK4/6 inhibitor plus fasting mimicking diet, we achieved the long-term control of tumor growth, and all the animals were treated with this triplet were alive at the end of the experiment. And in this specific tumor type, the mechanistic role of growth factors is due to the fact that the PI3 kinase AKT-mTOR pathway, which is activated by plasma membrane receptors that are activated by these extracellular growth factors, is a crucial mediator of primary or acquired endocrine resistance of these tumors. And so through calorie-restricted approaches, we are basically reducing in a non-pharmacological way the activation of this pathway. What about the immune system? So in syngeneic mouse models, it has been very nicely and clearly demonstrated that the immune system plays a role, that cyclic fasting mimicking diet in combination with chemotherapy promotes tumor infiltration by activated CD8 positive T cells, whereas the removal of these cells from the blood of animals almost completely reverses the anti-tumor effects of fasting mimicking diet in combination with chemotherapy. And this data have been confirmed in different tumor models, from breast cancer to melanoma to lung cancer models. You can see here in this a very nice paper published in 2020, the authors demonstrated that cyclic fasting synergizes with immunotherapy in murine lung cancer models that are intrinsically resistant to immunotherapy. And in this case, the anti-tumor effects and the immunomodulatory effects of fasting are mediated through the inhibition of the IGF-1 receptor pathway at the tumor level, so in cancer cells. And indeed, in this case, it was sufficient to administer recombinant IGF-1 to fasting animals to basically reverse the anti-cancer effects of fasting plus immune checkpoint inhibitors. So to conclude this first part, cyclic fasting and FMD have been shown to synergize with different standard of care anti-cancer treatments, including chemotherapy, immunotherapy, targeted therapies, and also other low toxicity drugs. This picture is taken from an article that is now, a review article that is now under revision. Don't look at details, but please believe me that in the vast majority of tumor types that have been used in preclinical studies published so far, fasting, with just a few exceptions, fasting and FMD showed potent anti-cancer effects in combination with systemic treatments. And then the molecular mechanisms that are responsible for calorie restriction-induced anti-cancer effects are highly heterogeneous, and they range between tumor cell autonomous anti-cancer effects, such as glucose growth factor restriction, or an enhanced production of radical oxygen species by cancer cells, to immune-mediated anti-cancer effects that are mostly mediated through an enhanced infiltration of the tumor by activated and memory CD8 positive T cells and by concomitant reduction of immune suppressive myeloid-derived suppressor cells. What about the clinical evidence? So in the next few slides, I will show you the results of clinical trials that we have been conducting in the last years. And for the conduction of these trials in 2016, we designed at the National Cancer Institute of Milan a sort of homemade fasting mimicking diet regimen, which, as you can see here, is a very low-calorie, carbohydrate and protein-restricted 5-day FMD regimen that we combined with standard anti-cancer treatments repeated every 3 or 4 weeks and followed by refeeding. And this FMD scheme was investigated in a first human phase 1b clinical trial that enrolled 101 patients. The primary objective of this study was to investigate the safety of this approach, in particular the incidence of grade 3 or grade 4 adverse events that were attributed to fasting mimicking diets, but we also analyzed a series of secondary biological and clinical endpoints. Of course, we excluded patients who were underweight or at high risk to become malnourished. And between January 2017 and January 2020, we enrolled 101 patients. The vast majority of them had breast cancer or other solid malignancies, but there were also patients with some leukemias or lymphomas. And this is the analysis related to the primary endpoint of the study. As you can see here, with an incidence of severe adverse events related to the FMD of 12.9%, this was a positive study because this percentage was lower than the pre-specified threshold of 20% that we fixed as sort of safety threshold based on which we would have decided whether to move on with additional clinical trials to investigate this approach. And this is the impact of cyclic FMD on patient body mass index. As you can see here, during each of 8 subsequent fasting mimicking diet cycles, of course, patients lost weight. On average, they lost between 3 and 5 kilos. But after the third fasting mimicking diet cycles, those patients who were able to continue the treatment did not undergo progressive loss of their weight and consequently of their BMI. And in terms of metabolic responses, we observed a very consistent and reproducible yet reversible decrease of blood glucose, insulin, IGF-1 levels, and this was paralleled by an increase of urinary and peripheral blood ketone bodies. So these metabolic results recapitulate the metabolic effects that have been consistently found to mediate the anti-tumor effects of fasting or fasting mimicking diets in preclinical models. And this metabolic response was very consistent across different tumor types in patients treated with chemotherapy or other concomitant treatments, and also regardless of tumor stage, advanced or localized tumors. But probably the most interesting biological signal that emerged from this study was the systemic and intratumor immunomodulatory effects. As you can see from these box plots that show the frequency of specific peripheral blood immune cell subsets that were quantified by flow cytometry analysis, we found that each FMD cycle was associated with significant downregulation of several populations of immune suppressive myeloid cells, both monocytic and granulocytic ones, and this was paralleled by an increase in the frequency of activated CD8 positive T cells and cytolytic NK cells. And in a few patients with a highly aggressive and deadly metastatic diseases, such as these 2 patients with advanced triple-negative breast cancer, we found that combining cyclic fasting mimicking diet with standard first-line chemotherapy resulted in complete and long-lasting tumor remissions. And more recently, we also published the results of this sub-analysis of the study, which we found that advanced TNBC patients receiving FMD plus chemotherapy had significantly longer overall survival when compared to a similar cohort of patients receiving first-line chemotherapy alone. But there were also patients with other tumor types who achieved complete and long-lasting tumor remission. This is the case of patients with extensive stage small cell lung carcinoma, which is a highly aggressive tumor, and these patients achieved complete and long-lasting tumor remission by combining a second-line immunotherapy with cyclic fasting mimicking diet. And 6 years after the initiation of the treatment, the patient is still alive and without any evidence of disease. Similar story applies to some patients with advanced pancreatic adenocarcinoma and KRAS mutated colorectal carcinoma. We collected these exceptional responders in a paper that we published in 2022. So this was, the previous slide showed that the immunomodulatory effect at the systemic level, but we also found at the tumor level, in a subset of 22 patients with early-stage breast cancer undergoing just one cycle of fast-mimicking diet without any concomitant anti-tumor treatment, we demonstrated that the FMD, 5-day FMD, boosts tumor infiltration by CD8 positive T cells, and by performing transcriptomic analysis and immune deconvolution analysis of transcriptomic data, in general, we found an enhanced tumor inflammation after fast-mimicking diet with several population of central and effector memory CD4 and CD8 positive T cells that were estimated to be enriched at the end of calorie restriction. And so we proposed this model of FMD-induced immunomodulation, according to which the reduction of myeloid-derived suppressor cell is more likely to depend on a reduction of blood glucose levels, whereas the reduction of growth factors and the modulation of some amino acids is probably responsible for the observed infiltration of and activation of CD8 positive T cells at the tumor level. What about metabolic interventions? There are a lot of metabolic drugs that are being investigated in cancer therapy. After the publication of this study by our colleagues at the European Institute of Oncology in Milan, our colleagues basically showed that combining glucose restriction with metformin, which is an antidiabetic compound that inhibits complex 1 in mitochondria, this combination results in a highly synergistic in vitro and in vivo anti-tumor effect in different tumor models. And so we went back to our original hypothesis that combining nutritional and pharmacological metabolic interventions may give rise to highly synergistic anti-cancer effects in cancer therapy. And based on this assumption and based on this preclinical data, we designed a phase 2 trial, which was the breakfast study, in which we randomized patients with early-stage triple-negative breast cancer to receive preoperative chemotherapy plus fast-mimicking diet or chemotherapy plus FMD plus metformin. So this paper is now under revision. I will show you unpublished results. So we enrolled this was a very small feasibility phase 2 trial in which we enrolled 30 patients randomized to FMD or FMD plus metformin plus chemotherapy. The compliance to the FMD in this case was really excellent, much better than in the previous study, with the vast majority of patients being able to complete the maximum number of allowed fast-mimicking diet cycles, which was 8. In terms of BMI modifications, we basically observed very similar results to the ones that we previously published in a much more heterogeneous course of patients. But this sub-analysis is especially interesting because we found that patients were overweight or obese at baseline after 6 months of preoperative chemotherapy plus fast-mimicking diet lost much more weight when compared to patients who were the normal weight at baseline. And we also analyzed computed tomography scans collected at baseline and at surgery, so after the treatment, to look at changes in different parameters that reflect total fat and muscle composition. And we basically found that all these parameters reflecting total subcutaneous and also visceral fat were reduced, but also the skeletal mass index was reduced after 6 months, but 1 year, after 1 year of follow-up, the skeletal muscle index was almost completely recovered. So very good news on this side. And in terms of anti-tumor activity with 56.6% of pathologic complete responses, which was the primary study endpoint, this study, which is of course not conclusive because of the lack of a control arm, but when compared to historical data, this percentage compares very well. And so in terms of clinical responses and also long-term event-free survival, this study is a very promising one. Then we performed the biological analysis on tumor samples collected at the baseline and after 1 treatment cycle. And these analyses were performed to looking for precocious biomarkers, especially metabolic biomarkers of tumor response to the treatment. And we found a significant and very precocious downregulation of several metabolic pathways related to glycolysis, pyruvate metabolism, and TC cycle. and these signals were only observed in a subset of patients achieving pathologic complete response at the end of the treatment. We also found that the glucose transporters GLUT1 and several TCA cycle enzymes were independently associated with an increased percentage, with an increased chance to achieve pathologic complete response after adjustment for other clinical parameters. And finally, by performing single-cell RNA sequencing analysis in baseline and after one treatment cycle tumor samples, this analysis was able to basically detect, as shown in this UMAP, not only cancer cells, but tumor-infiltrating immune cells and other population in the tumor microenvironment. What we found from this analysis in a subset of patients was that the most glycolytic cells, including cancer cells, myeloid cells, pericytes, and myoepithelial cells, underwent the strongest reduction of glycolytic metabolism during fast-mimicking diet only in patients undergoing pathologic complete response. So here we have the confirmation that actually cancer cells, among other glycolytic ones, are sensible to the fast-mimicking diet in terms of reduction of their glycolytic metabolism, specifically in the tumors that will be responding to the treatment. So I conclude this second part by saying that cyclic FMD is safe and feasible in cancer patients, provided that patients are managed in an appropriate way and by an expert team of oncologists and nutritionists, and that studies conducted so far, even with such a severely calorie-restricted approach, also showed the promising signals in terms of anti-tumor response and in some cases of a long-term patient survival. What about the future perspective? So after seeing the results of the breakfast trial, which basically showed that the FMD is associated with good responses, but metformin did not result in highly synergistic anti-tumor effects when combined to fast-mimicking diet, we have recently initiated a large randomized multi-center phase 2 trial, which is a breakfast 2, in which patients with early-stage TNBC are being randomized to preoperative standard chemoimmunotherapy versus standard chemoimmunotherapy in combination with fast-mimicking diet. So we are proceeding in this, for this study without metformin, but with the same 5-day FMD regimen that we have investigated so far. And this is the list of centers that will be participating in this trial. So to follow the patients in a centralized manner, we developed this web app, which has very nice interface and which will allow us to collect information about the patient diet, about quality of life, adverse events, and there is also a chat that we may have with the patient. There is also the possibility for the patient to activate an immediate phone call with the oncologist in case of severe adverse events. So we are trying to do the best to minimize adverse events and to make this approach broadly broadly expandable also in other institutions, in other centers. This is another small phase 2 clinical trial that we are conducting in patients with extensive stage small cell lung cancer. So based on a single patient who responded very well and was basically cured through this approach, we are now assigning patients not progressing after 4 cycles of induction chemoimmunotherapy to receive maintenance immune checkpoint inhibitor immunotherapy with atezolizumab in combination with 5-day fast-mimicking diet. This will be a small monocentric clinical trial in a population of patients characterized by a highly aggressive and deadly disease with an expected survival of less than 6 months after finishing the induction. And finally, based on results of preclinical studies that I showed you before, indicating that endocrine therapy plus CDK4/6 inhibitor has highly synergistic anti-cancer effects with fast-mimicking diet in preclinical models of hormone receptor-positive HER2-negative breast cancer, we are now going to initiate a second multicentric clinical trial in a totally different population of patients with hormone receptor-positive breast cancer and metastatic disease. And the primary endpoint of this trial will be 12-month progression-free survival. And this is a list of clinical trials that we have been initiating on that will initiate soon by continuing to explore our FMD, our 5-day FMD approach in different clinical contexts. So we will see whether these clinical trials or other trials that are being conducted in other European countries or in the US through different FMD approaches will provide sufficient evidence. All of these trials are phase 2 clinical trials. Hopefully, some of these approaches will provide sufficient evidence of enhanced anti-tumor activity or enhanced or improved the long-term clinical outcomes, and we will see if in the future there will be space for phase 3 clinical trials. So I would like to acknowledge a lot of people. First of all, the patients and their families who are taking part in these studies, or our preclinical and clinical groups, our internal and external collaborators, both scientists and clinicians who are helping us to design, conduct, and implement clinical trials with this approach in different clinical contexts, both in the early stage and in the metastatic disease. And of course, thank you very much for your attention.
Thank you very much. I will take you one question, if it is possible.
While that we can connect it, so you can. I'll just connect.
Okay. Do you have any question? Okay, thank you. Now, yes, Gökhan, we will connect to Gökhan Hotamişli via Zoom, and I would like to introduce Gökhan. He is the James Stevenson Simmons Professor of Genetic Metabolism in Department of Molecular Metabolism at Harvard T.A. Chen School of Public Health. Dr. Hotamişli graduated from Ankara Anadolu High School and Ankara Faculty of Medicine in 1986 with the title of medical doctor. After his residency, he completed his doctorate at the Harvard University Medical School in 1994 and established his own laboratory as an assistant professor in the Department of Nutrition and Biological Science at the Harvard Chen School of Public Health. In 2003, he received the title of professor and became the founding head of the Department of Genetic and Complex Disease. Hotamişli Lab at the Zafer Care Center at the Harvard School of Public Health studies innate adaptive pathways involved the metabolic health and disease such as obesity, diabetes, cardiovascular disease. For the past 25 years, his lab has made important contribution to the burgeoning field of the immunometabolism and metaflammation, studying the interactions between the metabolic and immune response as critical drivers of numerous chronic disorders. Gökhan? Dr. Hotamişli, do you hear us?
Yes, I can hear you.
Yeah. If you let me, I will address him in Turkish. Merhabalar. Sizi burada da ağırlamak istiyorduk ama böyle de olsa bizim için sizinle temas etmek bir onur. Öğrencilerimiz için de öyle, bizler için de öyle. Sizinle gurur duyuyoruz. Ben sizden konuşmanızı yapmanızı istiyoruz. Biraz zamanın gerisinde kaldık. Çok memnun olacağız sizi dinlemekten. Now I am introducing Dr. Hotamişli. As you know, he is from Harvard School of Public Health. We are proud of him, and I hope he will explain regulated subcellular architecture in fasting. Please, Dr. Hotamişli.
Çok teşekkürler. Merhaba hepinize Boston'dan. Ben de tabii kişisel olarak katılmayı çok arzu ediyordum ama maalesef program izin vermedi. Şimdiden hepinize başarılı bir konferans diliyorum ve bundan sonrasına İngilizce olarak devam edeceğim. Thank you very much for this kind introduction. Yeah, I wish I could be in person with you all there, but this time it wasn't possible. Especially, this is a very, very special gathering for me. So I want to thank Kocaeli University for hosting this, and President Zafer Cantürk to make the university campus available for this really wonderful occasion to remember our dear friend and colleague James Mitchell with science. Of course, these are difficult times for science in the world. Scientists are extremely stressed and anxious. I mean, campuses are facing tremendous challenges and difficulties. You know, truth and output of science is challenged as much as the quality of science it is produced. So it is really wonderful to bring scientists together and remember a fantastic scientist by talking about science. I know the time is late, so I will try to give you some tidbits of a story, and thanks again President Cantürk for the invitation and hosting this event for us. So yeah, I'll talk a little bit about fasting because this was one area Jay was really obsessed and made wonderful contributions, and that was, of course, I'm sure you heard already that one of the perhaps best established paradigms in medicine is the benefits of long-term caloric restriction. In some ways, maybe long-term fasting. I mean, although this has been known for a century, Jay was really, in my view, the first one to do two things. One is to really move this field from an observational intrigue to a tangible, testable, and molecularly explainable concept. And then the second, actually, he took actions to translate it in more practical, applicable, convenient manners. So, in remembering Jay, I talk a little bit about our recent work related to fasting. And I want to start by thanking our group. Of course, everything I say is the product of this wonderful group of young scientists that I had the pleasure of working together. And of course, some of them are with you today. I should thank them for organizing this meeting, Furkan Burak and Chris Hines. Thank you. So just to start the conversation, at least in our group, the view of health and disease comes from the interface between our biological infrastructure, the assets that we have at the biology level, and their adaptation to environmental input. So this input could be in many forms. It could be social stresses and inputs, ecological toxins and forces, and microbial and dietary input. So so when you, let's say, for the topic of the day, if the question is nutrition and diet, when a stress enters the system, then the body needs to launch an adaptive response, and so that this stress is mitigated and then handled productively. And if this is successful, then you maintain homeostasis and health. But if the stress supersedes the adaptive capacity, or adaptive capacity is compromised in some ways, then a mismatch occurs, and the homeostasis cannot be established, therefore you experience disease. So most of the things I'm going to talk about are in this axis, and our lab actually works in this two axes. One is the environmental input from diet, and the second is the environmental input, which is, it could be, of course, intrinsic or extrinsic in both cases, that controls metabolism on one hand, immune response on the other hand, to put together the greatest of all adaptive systems, called immunometabolism. And most of really our work in the past 30 years is really understanding immunometabolism. I mean, map up some of the pathways and then see if we can generate something useful for practical purposes. And we have two domains in our group. One really relates to the substrates and mostly lipids, which I am not going to talk about today. Maybe Furkan mentioned some work about our recent work on a new hormone involved in this aspect. But today I'm going to talk about structural homeostasis, a new concept in metabolic adaptation, and mostly focusing initially a little bit on the endoplasmic reticulum, but by using endoplasmic reticulum as the master of all subcellular organization, I will talk about overall structural programming of the cell in facing metabolic challenges. So this is just to give you a little background very quickly of 20 years of work, is that there is a peculiar observation in higher organisms when they face metabolic challenges, that it actually disrupts the function of organelles. And the chief among this is the endoplasmic reticulum. And in addition to its classic biology in folding, quality control, secretion, targeting of proteins, ER has specialized metabolic functions. And so we have recognized this in the early 2000s and really couldn't quite understand why a really powerful and strong organelle like endoplasmic reticulum fails. For example, when you're exposed to continuous caloric-rich diets or in the course of obesity. So this really, we don't really need to go through this cycle, but we've identified many molecules and signaling systems that disrupt the function of ER over the years. And most recently, we recognize there is also an architectural problem in the way ER is organizing its metabolic responses. And why, for example, to consider subcellular architecture as a determinant of function? Of course, I mean, from an architectural point of view, a form is really a requirement for function. I mean, if you want to cross the Bosphorus, you need to have a structure, a form, which is called the bridge. And so as long as there's a bridge, the function could be accomplished to cross from one side to the other. But in biology, there is a great deal of diversity in the subcellular structure. Unlike the higher organization, higher structures like organs or the whole organism, subcellular structure exhibits a far rich and pleiotropic diversity. And so I'll give you three reasons why subcellular structure may be important, just to establish the postulate. So one is, it's very different between cells. So if you look at multiple cells, so exocrine pancreas is full of ER because it needs to secrete liters of proteins, digestive enzymes every day. Liver is extremely pleiotropic, rich, diverse structure because it does many things at the both synthetic and disposal level, and energy and substrate supply for the whole organism. Whereas if you look at, for example, adipose tissue, in the middle you see brown adipose tissue, on the right you see white adipose tissue, very different. I mean, brown adipose tissue is very homogeneous with mitochondria, but very little endoplasmic reticulum or other things. Whereas in the white adipose tissue, you hardly have any subcellular organization. It's all squeezed to the edge. So if there is so much diversity, this might be a function of what these cells are supposed to do. Second, during the course of disease, these structures change. And this we have recognized in Anna and Benedicte's work in 2014. This is published, I'm not going to talk about, but in obesity, you see form is really exhibiting differences. And for example, more interactions between ER and mitochondria. And third, and more relevant to today's talk, normal physiology also regulates this subcellular structure. I'm all talking about liver cells right now, but in a normal, healthy animal, there is great level of difference between fed and fasted state of the hepatocytes. And I will come back to this towards the end of my talk, and just to show you first some of the analytical platforms. So, I mean, the question is, can we really interrogate the form-function relationship at the subcellular level in metabolism? And of course, we wanted to be able to do this in the native environment, not in cultured cells, but in the actual organ context where the cells reside. So for this, I need to acknowledge Ana Paula Arruda and Güneş Parlakgül. They're wonderful, wonderful, exceptional scientists with whom I worked for many years. Now they have their own independent group at UC Berkeley. And so with them, we decided to tackle this question, and then had a wonderful collaboration with Shan Shu and Harold Hess, who really developed fantastic optical systems to be able to look into tissue and in extreme detail. So with this, we explored liver tissue using focused ion beam scanning electron microscopy, a technique that can handle high volumes and extreme resolution at single nanometers level resolution of tissue architecture. And this is the first and the biggest example of application of that technology. So here, I'll just show you a quick few videos. You take a section, you pictures, and then burn the surface of that preparation with a very precise laser, and then continue to take pictures, tens of thousands of such pictures with single nanometer distance, and then you annotate this whole data to establish the three-dimensional structure of the inside of the cells. So this example here is published, so I'm passing this quickly, shows you a normal liver cells. Greens are ER, purples are mitochondria. ER is extremely, as you see, well organized in parallel sheets. It's a crowded space, but extremely well organized. However, if you do a comparative analysis, for example, between a lean, normal liver tissue and an obese liver tissue, you see the subcellular structure is almost unrecognizable in the course of obesity. So this entire organization of parallel sheets are lost, and the liver, of course, exhibits many other structural changes at the subcellular level. And so this project actually also brought us together with the media artist Refik Anadol, and then with whom we actually reanalyzed this 40 terabytes of imaging data, and a small scale is converted into this sort of visualization. I'm just sharing it to you so you have a sense of the organization of the membranes in a normal liver tissue. And in this imaging system, you can actually go inside of these structures and then visualize how the membranes relate to each other and how they are organized under different circumstances. And if we take this, for example, I mean, same approach and look at the obese liver, you will see that it's very different. It is full of lipid droplets, these yellow dots that you see, and the organization is completely disrupted. So the sheets are lost, yet they're replaced with this ER tubules. And of course, in this system, we can also go inside these tubules and then examine the surface properties, organizational properties, etc. So this first observation that the structure is so dramatically changed between lean and obese liver, of course, the question this begs is, does this have any relevance for function? And again, I will really skip this, if you forgive me, quickly, just to say that we can actually test this by trying to fix these structures. So can we, to establish causality, can we return the obese liver a little bit towards the lean liver organization, and what would be the consequence of this metabolically? So to do this, we used a membrane shaping protein, a synthetic construct that is taking advantage of the properties of this membrane-shaping protein called CLIMP63. And here you see two EM pictures from obese, extremely obese animals' liver tissue. On the left is the no intervention, on the right this synthetic intervention. You can see that the parallel sheets are appearing in this setting. And then these parallel sheets are actually very healthy because you can also detect ribosomes. They're studied with ribosomes, which is something only you see in the organ context. So if you do the same experiment in cultured cells, you will not see this healthy and relevant organization. So what happens if you fix the structures in this manner? Although the obesity remains at the same extent, extreme obesity, the liver starts functioning in a very healthy manner. So it reduces its glucose production. You see on the left, the animals tolerate a glucose challenge far better than the controls, and the liver actually starts disposing of its lipids, accumulated lipids. So just by a simple intervention to improve the structural, subcellular, architectural organization, you can change the metabolic program of a hepatocyte. And then if you do this in vivo, you can actually achieve improvement of liver function, metabolically speaking, in the context of extreme obesity. So this tells us that structural organization is a key in determining how a cell will decide what to do metabolically. So this is a new concept of achieving metabolic homeostasis through structural organization. So from this, I want to come to the last part of what I want to present with you. So of course we start with pathology, but I, like many of my colleagues in the field, strongly believe that most pathologies emerge from a process that is part of our normal physiology. So in the course of disease, whether it is stress or microbial input, our inherent physiological pathways are either diverted, hijacked, or exploited to serve or promote the disease state. So what is the healthy counterpart of what I mentioned to you, the structural, structure-driven metabolic program? So here I would go into fasting and first share with you, two sets of puzzling observations, one of which, of course, also has been a really topic of extreme debate, criticism in the field. So one is a study done by Sunan. Sunan was an extraordinary, really, mind, which we worked for many years. He then also moved on to build his own group, independent own group. But Sunan did a really, I think, foundational work to examine ER in different conditions and look into all of its constituents, all of the lipids, all of the proteins, membrane structures, but also isolated ribosomes that are associated with the ER, and looked at their translational profile. So when we did this experiment, and Sunan was really so bright that many of us understood what he was doing many years later, and sometimes actually still trying to do that. But this one he also couldn't understand, because what we found was if we look at the translational profile, so which proteins ER uses to translate in the obese state at the fed and fasted condition versus the lean state, again, fat and fasted. But what we found was obese liver at the fed state had the greatest resemblance to the lean liver, which was fasting. So this is very, very bizarre, and the correlation here is extremely high. The statistical significance of this is p 10 to the minus 12, and almost a linear relationship. But this, of course, is not lean fasting. So obese tissue seems to be trying to mimic in its adaptive response the lean fasting state, but unable to do so. And then, I mean, I told you obesity leads to enrichment of ER mitochondria interactions, or MAMs. And it turns out fasting also does the same thing. So why? So this was actually the basis of debate and criticism. So how come, you know, something that is part of fasting, which we generally consider as healthy, is also a feature of obesity, which we generally consider as unhealthy. So I will explain this in my remaining time. So of course, for this, we also decided to resort to the same technology, looking now into a different paradigm. So we decided to do a deep mapping of subcellular architecture in fed and fasted state. I mean, this time we also put some focus on mitochondria and distribution along the zones of hepatocyte. And actually, just this morning, like a few minutes ago, I received a note that this paper is now accepted finally after years of effort. So I want to congratulate Güneş and Anna once again. So just to give you an idea, I mean, what kind of analysis is this? So, I mean, usually people look at, you know, 5, 10, 20 mitochondria and reach some conclusions, which are, of course, more often than not, are incorrect. And so here, Anna and Güneş examined 108,000 mitochondria. So all of the mitochondria that we can actually fit into this data set. Again, it involves, I mean, 22 terabytes of imaging data generated from lean and obese animals at the fed and fasted states. The same paradigm, this time we're looking at structure. So, I mean, the first thing here to recognize is a great reorganization, really. So again, here on the left you see the fed state, on the right you see the fasted state, and we see a massive reorganization of the subcellular structures. And one thing I want to highlight, and I'll show another example of this, is what happens to mitochondria when an organism experiences fasting. So on the left you see this rather homogeneous shape and morphology of mitochondria at the fed state. But if you look at on the right, mitochondria comes in great structural diversity, dramatic changes in its structural organization. So here, for example, I can show you another example. So this image on the top left shows all of the mitochondria in the area that we can examine at the fed state, and underneath that at the fasted state. So from this picture you can gather that this is a very, very, very different morphological organization. And then in the lean animal, in a controlled normal animal, if you look at fed and fasted collection of mitochondrial shapes, you see a great diversity. So first thing we recognize is fasting has an enormous impact on mitochondrial morphology. And then the second is really the interaction of mitochondria and ER, subcellular membranes interactions. So here, for example, a case from obesity, a single mitochondria in the red, and a gray parts show endoplasmic reticulum. And you can see the relationship of this is very intimate. So ER is too close, almost sticking to the mitochondria. It's patchy, disorganized, and the mitochondrial shape is uniform. If you look at the same, for example, we take one example from fasting, the mitochondria shape is greatly altered. It's much larger, elongated, and the ER just wraps, envelopes the mitochondria. It's like a glove and hand, and a very interesting, a specific type of continuous, very well organized, distant, carefully arranged relationship between ER and mitochondria. And then the second thing we recognize that this interaction between ER and mitochondria is dependent on the zonation of the hepatocytes. So hepatocytes in the liver are organized generally in a gradient from portal region to the pericentral region. Of course, these cells, since portal area is rich in nutrients, and then there's a gradient, they showed differences, and there are also functional differences between the cells, for example, reside close to the portal area versus central area. And commensurate with that, the structural organization happens in the periportal and then in a reduced level in the mid-lobular area, but we see no structural organization in the pericentral area. So where the metabolism is critical for example, in this paradigm, you see the even differences between hepatocytes that are conducting the metabolic adaptation versus not. And I think here, for example, is a global picture of the same, zonation. If you look at the periportal area, you see great, so ER wrapping, enveloping the mitochondria, whereas in the pericentral area, you just see the parallel sheets and really standard interactions, rare interactions between ER and mitochondria. So the second thing that we recognize, and this is, I think, the most critical one. So I told you, obesity is a condition where ER and mitochondria interacts a lot, whereas fasting is also a condition ER and mitochondria interacts. But if you look at, the devil is always in the detail. So if you look at this in detail and much more carefully, yes, they both interact, but the interaction is very different. So on the left, you see obesity. So in obesity, the part of ER that interacts with mitochondria is the smooth ER. So it's a very, as I mentioned to you, it's a very tight and uniform interaction, and this interaction can be measured to be all in 0 to 25 nanometer distance. Whereas if you look at fasting, I don't really need to even describe this to you. First of all, different part of ER interacts with mitochondria. This is the rough ER this time, and then structurally it's continuous, uniform, and it is studded with ribosomes. So a very different kind of interaction between ER and mitochondria. So let me finish by saying, what does this mean? So there is complete restructuring of subcellular architecture in the fed and fasted states. Here's really a dramatic visualization of that. And so what does this mean? And so to do this, we again resorted to membrane-shaping proteins and then generated some synthetic construct, delivered them into liver one by one to ask, can we find what is responsible for this organization? And for that, one little screening trick that Anna and Giresh used was to look into all known membrane-shaping proteins and determine which one actually shows zonation. So the idea is if something is zonated and also regulated, for example, in obesity or fasting, this is a more likely candidate. And then this analysis actually brought us another membrane-shaping protein called RBP1 or P180 as a potential candidate. And then when they deleted this protein from the liver, we recognized that the liver now, in the absence of this protein, can no longer assume the shape that it does during fasting. So fasting-related ER mitochondria interactions are dependent on the presence of this protein. And not just the structure, also in the absence of this protein, for example, liver has difficulty in conducting fatty acid oxidation. So in the cells and in the animals, we see higher accumulation of lipids in the absence of this protein, and the oxidative capacity is reduced. If we do the opposite, if we reintroduce this protein into the liver, then the oxidative capacity increases and the lipid accumulation decreases in this liver. Again, just by allowing the subcellular architecture to resume its proper shape in the course of fasting or disease state, we can generate a solution for its functional problems. So let me wrap up, just giving some, I mean, I mentioned all of this, but key take-home messages from this level of analysis that we have learned so far. One is subcellular architecture is dynamically regulated. This is true in the case of fasting, but also regulated in slightly different ways in obesity. The structural adaptation is zonated in the liver. And this zonation and the structural adaptation is required for proper metabolic programming to meet the demands of the organism. So the really interesting part is in the disease, it seems like the liver, at least in the liver, the hepatocyte is locked into a rigid structural form. So it cannot change, it doesn't respond, and cannot therefore carry out adaptive changes during fasting. So taken together, I think I want to put forward a thought that subcellular architectural regulation is a key component of metabolic fitness and flexibility. Perhaps this is a new way of building homeostasis, and the metabolic state demands the cells to assume a certain shape so that they can conduct their function. So with this, I want to finish and thank you, first of all, for inviting me, being part of this wonderful event, and listening to my talk. So I want to make a quotation to Yunus Emre. It says, whatever separates you from truth, just throw it away. It will disappear anyway. So don't be discouraged, please, with the challenges that science presents. It presents more rewards than challenges in the end. And I want to say salute to my really dear friend, Jay Mitchell. So this is a picture, really very special moment in our lives, also in Jay's life and, of course, his family. This was the day Jay was promoted to professorial level and achieved tenure at Harvard. And then as I did at the time for our really brilliant faculty, me and my wife took them to a very nice dinner in our favorite restaurant in Boston. So his memory really lives with us. In his short life, Jay left us so many treasures, so many human treasures, so many scholarly treasures that we continue to remember and miss, and still haven't found a way to fill that gap. So thank you for remembering and helping us to remember our really great friend, Jay. Thank you.
Thank you very much, Gökhan. It was great presentation. It is really excellent presentation. Thank you very much. Is there any question from audience?
Hocam, I would like to say something.
Yeah.
And Gökhan Hocam, Furkan here. So the work that you showed now, even from the publication, so one medical student here came all the way from Hatay, from the earthquake region. Demet, I want you to come here, please. So she basically painted. I just received this. So she did this painting and came today all the way from Hatay to Kocaeli just to get all the things. So here, so I just want to thank her, and then, you know, I want you to know that you inspire all the young generations. You inspired me when I was a student here, but it just continues. And then, so I was like, it's incredible that, you know, how they were aware of all of this development and made this painting. So thank you so much again, and I want you to show Gökhan Hoca and everybody.
Merhaba, çok teşekkürler. Ne kadar güzel. Çok memnun oldum bunu duyduğuma. I'm so happy to see this painting and hear your interest.
I'm bringing it to Boston. Thank you.
Thank you, Furkan.
Congratulations. Thank you very much. Gökhan, thank you very much. Please say our best wishes to your family
Thank you.
and your
Thank you.
colleagues. Thank you very much for being us. Bye-bye.
Thank you. Goodbye. Good night. Bye-bye.